All right, sir.
Can you see my
Good
screen?
evening, everyone. Good evening,
Yes,
everyone.
yes, sir.
So today we are having a very bright personality of the pharmacy profession, Dr. Farhan Jalees Ahmed. Uh, Dr. Farhan is currently working as a dean in the School of the Pharmaceutical Science and Research in Jamia Hamdard University. Dr. Ahmed has a rich experience in the industrial research through his fruitful association with the Randaxi Research Laboratory for 6 long years. His domain of the research theme is including the development, scale-up technique, technology transfer of the nano delivery systems. Currently, Dr. Farhan Ahmed is working on the many projects which are funded by the DPT, CCRUM, Ayush, UGC, DST, and SERB. Without wasting time, I would like to invite Dr. Farhan Ahmed uh, sharing the view on the nanotechnology based intervention in the phytoconstituents formulation. Welcome, sir, in the ISF series of lecture. Over to you, sir.
Thank you so much, Dr. Nitin. First of all, on the outset, I want to thank your platform for sharing me and giving me this opportunity to share my research on this wonderful platform where I can connect with large number of audience from pharmacy world. So today, the topic which I have chosen is about the work which we have done in the last 10, 15 years, where we are trying to amalgamate the modern technology like nanotechnology with our traditional medicine and taking the cue from the work which is done by our hakims or our Veds, where they have established a lot of herbs as a successful treatment of many, many ailments. Although in the absence of those modern analytical techniques, with their experience, with centuries of experience, they have identified certain herbs which shows a lot of potential in many diseases. So today I'm going to talk about how we have used nanotechnology to improve the therapeutic effect, pharmacokinetic and pharmacodynamic of these excipients. First of all, I'll just give you a brief introduction about my university, which is Hamdard University. It was started in 1968 when Hakim Abdul Habib set up an Institute of History of Medicine and Medical Research in Delhi. In 1963, he started Hamdard Tibbi College, which is Unani College. In 64, he established Hamdard National Foundation. This foundation was established from the earning of Hamdard labs, and he dedicated all the earning of his company into this foundation. And this foundation was with the objective of developing many institutes, schools for weaker section of the society. In 1989, his dream was fulfilled when our university got the status of deemed university by Ministry of HRD. So these are a few images of our facilities, our institutes, our convention center. We have students from more than 30 countries, and these are the few research labs we are working. Our students are working. So if you look at our ancient system of medicine, two major systems which are in practice in India is Unani system of medicine, which comes with Mughals as a Greco-Arab system of medicine, and then our own system, which is Ayurvedic system of medicine. In these systems, whatever herbs we are using, these herbs were formulated in different forms, either in the form of ashthas or in the form of churnas. So they have very, very specific forms. But now in today's world, we are trying to use those herbs and their pure constituents. So these pure constituents, when we look at their availability in pure form, is a challenge. Although they are available in the market, but they're not available in bulk scale. And when they are available, there is a lot of variation with respect to their solubility, with respect to their stability, with respect to their crystal form. In compared with modern medicine, these things are not available. Then they have a very big challenge about their solubility. If you look at all modern or all popular herbs like curcuma longa or saffron or thymoquinone or resveratrol, they are highly insoluble, and because of this, their absorption is also limited, and which affects the pharmacokinetic of these drugs. If you want to take them orally, their pharmacokinetic is very poor. Then stability. Now, if you look at the stability of modern medicine, all the stability data is available in the literature. But for these herbs, this type of data is not available. You have to generate the data. You should know what is the solution stability, what is the solid state stability, how it is going to behave with the excipients. So this information is not available. Then because of the variability of the source, the reproducer of the result is also highly variable. If you are taking a curcumin from one source, and if it is not available in the large quantity, if you change the source, its results are also changing. Now, what we do is we try to apply nanotechnology to these excipients so as to improve their solubility as well as their pharmacokinetic. We use the method of nanoprecipitation, or we use the methods of emulsification evaporation. In nanoprecipitation, the drug was dispersed in a solvent, and then with the help of sulfur stabilizer, it is dispersed and it is crystallized slowly in a controlled manner. And then it is passed to a high pressure homogenization to get the desired particle size, where we can arrest the particle growth by applying anti-solvent technology. In emulsion evaporation, we are using a polymer with organic solvent where we are dissolving the drug, and then try to control the solubility by removing the organic phase by rotary evaporation and then controlling the particle size. Then we can also use polymerization by where we are making reverse micelles of NIPAM or any other acrylic monomers, and these monomers are made into polymer by using methyl bisacrylamide, and reverse micelles are formed. These micelles can encapsulate insoluble molecule, and outer surface being hydrophilic, they become soluble in the aqueous media. This is the scheme where we can use NIPAM, vinyl pyridone, ambient water, and it can be polymerized to get the micelles. Then we also use nanoemulsion technology, where you can disperse the drug which are insoluble in oil phase, and it can be emulsifying with the help of surfactants and co-surfactants to get a globule size of less than 100 nanometer, which can have better solubility and better absorbability. We can use ternary phase diagram by calculating the soluble phase and insoluble phase, and we can absolutely have a range of oil, water, surfactants, and co-surfactants. Now, first example which we have successfully converted into a nano formulation, we have taken thymoquinone. Thymoquinone is very famous, which is obtained from Nigella sativa, and it is reported to be a very good antioxidants, anti-inflammatory activity. So this we have converted into a soluble form of emulsion, and where the particle size was ranging between 100 to 200 nanometer. And in this case, when we converted into emulsion form and we compared in the animal models, you can see the difference in overall viability. The percentage which is released is much higher as compared to the drug per se. When we compare the bioavailability, you can see the difference in bioavailability, where pharmacokinetic profile of the nanoemulsion is much higher as compared to the drug per se. And then when we compare the animal model, we also look at the safety part, where formulation treated part is also same as the normal. And you can, if you look at the biochemical enzyme activity, SGOT, SGPT, it has very good effect on these biochemicals in the liver action. Now, another example is saffronol. It is an aldehyde which is obtained from saffron, and this is also reported to have a very good effect on various behavioral issues. And we take a cue from the Ayurveda, where in case of a person if suffering from epilepsy, he was supposed to inhale the smoke. Now, smoke is also a colloidal dispersion of particles. So instead of smoke, we thought we will develop the nanoemulsion, and we will give it by nasal route. So when we prepared the formulation, we compared the saffronol in intranasal, peroral, and intraperitoneal. And if you look at this results, the brain uptake is higher when it is given by nasal route. And we prepare two formulations. One is saffronol nanoemulsion, as saffronol mucoadhesive nanoemulsion, and mucoadhesive nanoemulsion have the similar particle size as nanoemulsion, and we compared them in the animal model. If you look at the in vivo gamma counting studies, where we tag the nanoemulsion with technetium, and we give it by intranasal route, normal saffronol and saffronol emulsion, saffronol mucoadhesive nanoemulsion, and by IV route. And if you look at the uptake, in this case, if you take the brain uptake, it is highest with mucoadhesive nanoemulsion. So this completely confirms that if we are giving by nasal route in the mucoadhesive manner, it can be absorbed and cross the barrier and reach the blood size. If you look at the brain to blood ratio, it is highest with respect to formulation which is mucoadhesive in the form of nanoemulsion when it is given by intranasal route. These things are also confirmed by animal studies, where we perform the PTZ model as well as ICS model. In both the models of epilepsy, if you look at that, compare the activity of normal saline with our emulsion. And if you see, the latency to PTZ induced myoclonic jerks is highest when it is given in the mucoadhesive form. Similarly, here latency to PTZ induced generalized clonic seizures also, it has increased very significantly when compared to normal drug per se. Similarly, in ICS model, increasing current electroshock model, if you see when we are comparing with normal saline, sesame oil as negative and positive control, if you see nanomulsion and mucoadhesive nanomulsion showing the highest latency, highest threshold. Similarly, here also it is showing the same results. And we also compared the, because we are giving by nasal route is the most sensitive part, so there should not be any toxicity. So in order to look at the irritation, whenever there is irritation, there will be increase in mucosal surface temperature. So we measure the mucosal surface temperature by IR camera, and we found there is no irritation, there is no increase in surface temperature. And we also did the histopathology just to see the effect of these formulation on the nasal mucosa, and we found that it is very safe. Similarly, in the rotor-root experiment also, it has shown safety. Now, other work which we have done, we have tried to give in curcumin in the form of polymeric micelles, and we see if we can cross the blood-brain barrier. In the rabbit model, when it is given, these are the gamma images of the rabbit where a technician is tagged with the formulation. And if you see the left side image, you can see the brain image is much clearer as compared to the right side image, because in this case it is given in the micellar form with polysorbate 80 coded micelles, where it is given the normal form. So nothing is going to the brain, everything is going to liver, kidney, and then it is excreted. But we are here, a lot of drug has gone to the brain. So this has completely confirms that we could target the drug and we could cross the blood-brain barrier. Similarly, we have tested the same formulation in the Macau model, which is middle cerebral artery occlusion model, in which it is given in the, this is the positive control, where you can see the brain slices, it is red, there is no occlusion. But in this case, when we created the occlusion by using a Teflon wire, you can see the infarct or the which is created here. But when we give the drug in 3 different doses, curcumin is given at a dose of 1000 nanogram per kg, 10,000 nanogram per kg, and 1 lakh nanogram per kg. If you see, there is a complete reversal of the infarct, and the blockage is removed, and you can see that the healthy tissue is reappearing. So which clearly shows in the nanoform, it has very good effect on the brain ischemia. This is confirmed by our biochemical estimation of GST, GPX, GSS, and GR, where you can see the biochemicals, they are improving once the drug was given in the nano formulation. Similarly, here also, this is the effect of curcumin, SOD, catalase enzyme, grip test, rotor-root test, they all confirms our images. Now, other work which we have done with herbal as well as modern medicine, we try to target them to the lungs. Now, if you look at the dry powder inhalers which are available in the market, they are in the micron size range, and when they are given in the form of DPI, their lung deposition is reported in many clinical studies about 30 to 40%. So we thought we try to design the particles which are respirable and in the nano size range. These particles were designed by spray drying, so we can scale it up easily. And these particles, when we convert it into nano DPIs, their lung deposition is increased. If you compare this technetium image in the micron size range, if you see the particles, they entered the lungs and they stayed there for about a few hours. And after 6 hours, if you look at this image, all the particles they are gone from the lungs. But when they are given in the nano size range, the lung images are much brighter. It has gone deep into the lungs to the alveoli region, and even after 6 hours, the effect is remained. So a single dose of our nano DPI, it can act as a depot, and it can have prolonged effect inside the lungs. This technology we have used to develop the treatment of chronic obstructive pulmonary disease, which is a problem when you are working at high altitude. At high altitude, your lungs can swell, mucus can be accumulated, and your alveoli becomes blocked, and you have difficulty in breathing. Sometimes your death can happen during sleep also. So with a single puff of this formulation, you can see this is the actual image of the patient, where you can see the lung image is much brighter when it is given in the DPI form. And this technology can be used for the treatment of lung carcinoma, chronic obstructive pulmonary disease, pulmonary hypertension, asthma, tuberculosis. This is what we have done. This is the data which we have obtained from a patient in a base hospital of the defense, where the patients with proven pulmonary hypertension, we measured the baseline parameters, and single dose of DPI was given. So when it was given, the pulmonary arterial pressure is reduced by 25%, oxygen content of the patient is increased by about 10%, heart rate is reduced by about 8%, exercise capacity is also improved by 33%. This is a very, very important parameter, because if you are working at high altitude, you have a difficulty in breathing, your working capacity is reduced. But if you, with a single pump of DPI, if you can increase the working capacity, you can save the life of the person, where oxygen content is increased, and you can bring him down to the base hospital, where he can give a proper treatment. This is the products which we have developed. This technology, we have developed antidotes for toxic substances, which can be inhaled by a person when it is exposed to toxic substances. We have also developed antidote for organic phosphorus poisoning. We have also developed various products. So these products are basically are designed to target the lungs. So in this work, what we have done, we have converted allopathic drugs as well as herbal components. The main problem which we have faced with herbal components, you have to be very sure about the safety aspects of those herbal components, because lung and your nasal mucosa is a very, very sensitive organ. Its main function is to provide oxygen to the body. But if you want to use this organ for delivery, you have to be very careful. The choice of excipients, the choice of drug has to be very, very selective, because any excipient, any drug can start an allergic reaction, and then you have difficulty in breathing, and you have a lot of mucus, and it can goes into the allergic reaction, anaphylactic shock also. So those things you have to be very careful. When we are dealing with herbal components, the main problem with herbal components is their solubility and their absorption. So students can work on this aspect, because there are a lot of drugs or herbs which are used by ancient Ayurveda or in ancient Unani. You can take cue from that literature. I think we should read those literature, although we are pharmacy students, but we should also take knowledge from our history, and those things can be converted into modern form. Those things can be improved by using the technology, either it is analytical technology or it is formulation technology, which is available, and it can be used. So you can choose your topic of research by selecting any area of, you can say, gray area, where the treatment is not available in modern medicine. You can look into the ancient literature, and if you have found a treatment, that treatment can be improved, can be modernized, and can be validated, because these treatments, they are available for centuries. They are safe. But if you want to bring them to the market, if you want to bring them to the masses, you have to generate the data, which is not available. Either you have formulation data, preclinical data, clinical data, and then only you can successfully take it to the market, and it can benefit the large population of patients. So I think students can look at those literature. There are a lot of herbs, lot of excipients which are available, and more than 90% of these excipients, they are very poorly soluble. And if you want to develop a oral formulation, you have absolutely you have to improve its solubility, you have to improve its absorption, or you can target it to a specific organ. Targeting is also another area where students can select these molecules and target to a specific area, like if you want to target to the brain, or if you want to target to the cardiac tissue, or if you want to target to the liver, you can use your pharmaceutical or targeting technology, and you can either you can make micelles formulation, you can make lipidic nanocarriers, you can make localized depots. So those things, avenues are open to you. So whenever you are choosing research topic, you can instead of looking only at the modern medicine, you can look at your ancient literature and see where you can pick up a molecule, or where you can pick up a herb, where you can modify it, you can improve its pharmacokinetic parameter, eventually which will improve the pharmacodynamic. And if you can improve the pharmacodynamic, or if you can reduce the toxicity, your job is done. So with this message, I think I have shared the work which we have done. We have developed nanocollagen formulations, which are basically we are developed for the Australian company, Caltech Australia, where it is being used to improve the skin structure. It is used in the cosmetic preparation. We have developed nanodental formulations, which are basically for orthodontal infections. We have developed herbal formulations, where we have used various herbal constituents. We have developed nano formulations of fish oil, nano DPIs, and all. So these are the things which I want to share for this short presentation. If you have any query, you can write to me. You can have my email address, or you can ask question on this platform as well.
Thank you, Farhan sir, for sharing your research on this wonderful topic, because Ayurveda and our ancient literature are the demand today, after specifically after this COVID pandemics. So there are some limitation in the modern therapy. For that solution, we are look up to this herbal or the Unani kind of the literature. But at the same time, these Ayurvedic medicines are having some specific limitation in terms of the bioavailability, which we can minimize with the help of this novel tool and technology of the pharmaceutics. So thank you very much, sir, and letting the students and letting the whole pharmaceutical community and sharing your view on this vibrant topic. Now, I would like to invite our director, sir, Dr. G.D. Gupta, sir, for the conclusive remark.
So thank you very much, Dr. Nitinji. So good evening, sir. So
Good
first of
evening.
all, I am thankful, sir, for accepting our invitation, and you have given very important talk. This is the very related to the herbal as well as the nanotechnology, and we all know that during the COVID, everybody they are sufferer and they have used lot of utility of the herbal dosage form. So now this is the very important and applicable to the all, and most probably shootics person they are also shifted from the nanotechnology and using so many product related to the herbal formulation. So from core of my hearts and from ISF dialogue series as well as from ISF College of Pharmacy, I am thankful to you, sir, for accepting our invitation, and you have give a very technical and scientific talk, and your this valuable information will be useful to the or young scientists, those who are the working in this field. So I know that this time it is very difficult to the person to deliver or to accept the invitation like this time. So such types of the dedication to you, and you have accept invitation, and even in the off hours you have delivered the lecture for ISF dialogue series. So thank you very much. Thank you for the coordinator of IQAC. Thank you today's coordinator Dr. Nitinji to provide the opportunity to interact such types of the very dynamic and very renowned professional in the field of pharmacy. So thank you very much, Dr. Pran sir.
Thank you, sir. Thank you so much. And I think it was an opportunity for me also to be on this platform, where it can be seen by large number of students, and they can get benefited, and they can also take a cue from the work which we have